I am Professor A. Rathod, Department of Genetics and Plant Breeding, KKY College of Agriculture, Nasik. Today we discuss the syllabus of our course. The course name, that is the GPB 232, course credit, that is the 1+1, and the title of that the course, that is the fundamental of that the plant breeding. In that the first lecture we have to we leads to discuss the syllabus of our theory lectures or the theory topics. In that the first lecture, that is the definition, history of plant breeding, aim and general objective of plant breeding, then landmarks of plant breeding, Indian plant breeders, general objective of plant breeding, major achievements and future prospects. So in that the first lecture we have to study the whole brief introduction about the our plant breeding definition and their objective. And that the second lecture we discuss on that the self in complementarity. In that the third lecture we discuss about the male sterility, the definition, classification, type, genetic male sterility, thermosensitive genetic male sterility, photosensitive genetic male sterility, transgenic male sterility, cytoplasmic male sterility, cytoplasmic genetic male sterility, and chemical hybridizing agents. So it leads to 6% weightage, that is near about 2 or 3 questions there arise on that particular topic. Then the lecture number 4, that is the heritability. In that the heritability we have to learn from that their ancestor to the current one, what is the particular history of that the particular that topic. In that the definition, there are the two types, narrow and broad sense heritability, their components, that is the classification, definition of and features of the additive, dominance and the status, or and the one additional topic, that is the gene action. Then the lecture number 5, in that the concept of population genetics, definition and concept of population genetics, random mating, population, gene and genotype frequency. And there is the one important law in the to calculate the genetic constitution of that the particular plants. That law is, that is the Hardy-Wingman law. These are the two different scientists, they have to work on that particular gene frequency and they improve their law. The lecture number 6, that is the breeding methods in cell pollinated crops. In that our course, the 2 or 3 different types of pollination: self pollination, cross pollination and open cross pollination. In that the for development or improvement in that the self pollinated crops, we have to use the following different breeding methods. In that the plant introduction, pure line, then population, that is the pedigree method, bulk methods, back cross methods. Then the different breeding methods used for the cross pollinated crops improvement. In that the recurrent selection, it is the most important. Then the next topic, that is the hybridizing techniques. In that the what is the definition of hybridization, which are the different techniques and the steps we have to use in that the hybridization programs. In that there another topic in that the wide hybridization, in that their definition, types, main features, interspecific, intergeneric hybridization, these are their two main types. Then their examples is there. And the lecture number 13 is that composite and synthetic varieties. It is also the most important point in respect to that day plant breeding, their definition, step for development of composite and synthetic varieties, procedure of developing composite and synthetic varieties, its merits and their achievements. The lecture number 14, that is the breeding methods in sexually propagated crops. In that the most important breeding method, that is the clonal selection. It also will lead in that the upcoming lectures. In the what is the hybridization, their step, procedure, as like the sexual means in that the asexual also we have to follow the proper procedure for that the development of any variety. Lecture number 15, that is the mutation breeding and the polyploidy. It also the most important in respect to our course. In that the we learn their definition, their procedure, their advantage, disadvantage, and the last topic, that is the heterosis. In that heterosis, it is the superiority of the DF1 over its both the parents. Those parents we have to use for the crossing. On that parents, if their offspring is better, at that time we say it is the heterosis. Their types, then genetic basis or theories of heterosis, that is the inbreeding depression. So all over in that the briefs we have discussed the syllabus of our course. I hope you all very well learn about that the syllabus. Thank you.